Hello, I'm Stephen Lee, and welcome to Omics Quest. In today's Inside 10x Genomics video, we're diving into how single-cell 5' gene expression and VDJ libraries are made. These provide researchers with a powerful tool to study both gene expression profiling and immune profiling from the same set of single cells. Let's break it down. This video assumes you have a basic understanding of 10x single-cell technologies. Briefly, you start with gel beads, use microfluidics to create gel beads in emulsion or gems, synthesize cDNA inside each gem, then pull the gems to amplify the cDNA, and generate sequencing-ready libraries. Let's take a closer look at what happens after a gem is created. Inside a gem, there is a gel bead and mRNA molecules released from a cell or nucleus after lysis. To keep things simple, let's focus on the cDNA synthesis of a single mRNA molecule. Here's the sequence structure found on a gel bead. Now we have an mRNA molecule of interest. From here, reverse transcription takes over using a template switch oligo or TSO to generate the first full-length cDNA strand. I won't go into details here because I already have another Omics Quest video called Quick Guide to cDNA Library Construction and Template Switching Oligos, as shown here. Check out that video if you're interested in learning more about how template switching works. At this point, I want to emphasize that all of this is still occurring within individual gems. Once the first strand of cDNA synthesis is complete, the gems are broken and the cDNA strands are pulled together for further processing. After pulling, the cDNA strands undergo PCR amplification to generate sufficient material for library construction as shown here. From the same amplified cDNA, we can proceed to 5' gene expression libraries, VDJ libraries, or both as separate experiments. Let's first take a closer look at the formal. Amplified cDNA undergoes enzymatic fragmentation and size selection to optimize the amplicon size, as full-length cDNA is too long for next-generation sequencing. So even though full-length cDNA is generated, only the 5' end will be sequenced for gene expression profiling. Next, we follow the rest of the steps as shown here. I won't go into the details because I already have another Omics Quest video called Inside 10x Genomics, single-cell 3' gene expression libraries, as shown here. Check out that video if you're interested in learning more about how to go from full-length cDNA to the final 5' gene expression libraries. Now, let's take a close look at how we generate VDJ libraries. We can do this for B cells, T cells, or both as separate experiments. Their workflows are essentially identical. We first need to amplify cDNA molecules that contain the VDJ regions. To do this, we use primers targeting the read 1 sequence as the forward primer and the distal position of the TCR or IG constant regions as the reverse primer. We then use the same forward primer and a proximal position in the constant regions as the reverse primer to be more selective and amplify those molecules with VDJ regions. This hemi-nested PCR balances sensitivity and specificity. Next, we proceed to enzymatic fragmentation. This produces overlapping short reads across the full VDJ region, enabling accurate clonotype identification and quantification, i.e. TCR alpha and beta chains for T cells and IG heavy and light chains for B cells. The remaining steps are followed to generate sequencing-ready libraries. And here's the end result. We've generated a repertoire of cDNA molecules and used it to prepare both gene expression and immune profiling libraries from the same set of single cells. Thanks for watching Omics Quest. If you enjoyed this video, please like and subscribe to the channel.